Hello, I'm Babalola presenting investigating sex-related phenotype changes in knockout mice by applying deep learning to X-ray images on behalf of our group at the European Bioinformatics Institute. So we're part of a larger consortium called the International Mouse Phenotyping Consortium, and we carry out high throughput experiments with the aim of finding out what each knockout protein coding gene does in the mouse. So in particular, we're trying to associate phenotypes to each gene, and we do that by carrying out standardized procedures which require the measurement of 163 parameters on each mouse and finding phenotypes in knockout lines. Those are lines where we have taken out the particular gene we're interested in looking out, and we compare those with wild type mice, which are our controls, which have all the genes present. And this is just an example of going through a procedure. So we have the parameters for the procedure. In this case, you can see we've got the number of ribs on the right, number of ribs on the left, shape of the ribs, etc. And we get numbers for these from the images of X-rays, and then we pass those through our statistical machine, and that lets us know if it thinks that there is a difference by looking at the wild type controls and then the values for the males and females of the knockouts. And from there, if we think there is something of significance, we associate those with ontology terms that are internationally agreed, and we contribute that to the body of knowledge that is trying to build a picture of the mouse genome. So we're interested in deep learning because we have a lot of images. At the moment, we have to date over 500,000 images, and we've only been through just over 6,000 out of 20,000 genes. So this large number of images means that manual review that we've been using has not scaled well, and given the fact that we have a large number of properly annotated images and associated metadata, we are looking to use deep learning, in particular convolutional neural networks, which are ideal for working with images, and also because of the way in which they are built, the multiple levels of representation, which have lower level image information at the start of the filters, and as you progress through the network, the later layers have more higher level representations, which are meaningful to human beings. So those things are important to us, and also the fact that we can use transfer learning in making a model. So the next few slides, the next couple of 2 or 3 slides, are just going to go through some very brief details of neural networks for those people that may not be familiar with it. So in here we are showing 5 layers, and the first layer is called, so this is one of the simplest types of neural networks, the multi-layer perceptron, and the first layer is basically the input. In this case, it's just a vector of data, which could be vectorized images, or it could just be normal data, and each data point is associated with a weight, and we have what we call the hidden layer here, and here the weight, you get a weighted combination of each of your inputs with the associated weight, and you pass that weighted combination through an activation function, and then you get a result here, and this result is compared to what you would expect from your training set, so you know the answers already in advance from the training set, and you can see the difference, and using that difference you can go backwards, a process called backpropagation of the error here, and you can use that to update the weights, and then you go back forward propagation, and hopefully that minimizes the errors, and you go backwards and forwards until you get suitable values for the weights that has reduced your errors to a suitable amount. And using those weights, you can now apply them to new data to predict what the output will be for unseen data. Just two other things to point out. It's loosely based on the human nervous system, and also this hidden layer is the number of hidden layers you have is what gives the term to deep when we talk about deep learning. So you have many hidden layers, which you'll see in the next couple of slides. So we are interested in convolutional neural network, and this slide just talks about convolution briefly. So this is a matrix or a filter with values in it, which are called weights. And again, similar to what we did in the previous layer, this is our input image. And if we superimpose our filter on top of the image and we apply the weights to the values in the image, we can compute a number. And if we move it across, we can compute numbers for each part of the image that's covered, and that gives you so we take the image and we get a convolutional feature from it. Now the numbers in here are our weights that need to be trained by our forward and backwards propagation, and in doing that we create weights that extract meaningful information that at the end point of the convolutional neural network helps us to get the output that's similar to that we have from our training data. And this is just a visualization, just an example to show the application of a filter to a real image. And in this case, this is actually a handcrafted filter that's been used in computer vision for many years, called the Sobel filter, which looks at differences in the y direction vertically, and you apply that to the image of the cat, and as you can see, it highlights places where there is a difference in the vertical direction of image pixels. So going on forwards, the idea of applying convolutional filters to images is not new, and we have already many handcrafted filters, but in the CNNs, the data is what determines the values of the weights, and also we are not limited to 3x3 filters. We could have different sizes of filters, and in the ways they are configured in the hierarchical organization, typically people have a low number of filters at the start, and then the number of filters, this last number represents the layers of filters. So you have 64, 128, 25, 65, 12, and that gets bigger. But the actual dimensions, the XY dimensions of the images in the 2D case gets smaller by a process called max pooling layers, which just shrinks it in this case by half as you go through each max pooling layer. And then at the end you get the normal fully connected layers of the CNN. So the important thing to note is that at the starting layers, low-level image features are extracted. However, as you go towards the later layers, these are combined to higher-level features that are more meaningful to human beings. And then there are methods that allow us to visualize the inner workings of the CNN and seeing what is important in these features. So for our particular case, we built a CNN to classify images as males or females, and we use transfer learning, whereby an image CNN that has been trained on a different data set, usually a more larger general data set, is applied in another case. And in our case, the images were trained, VGG16, which is the model we're using, was trained on a data set called the ImageNet data set that had over 15 million images, although just a subset of those images were used to train it. And in this case, we had 1,000 categories used in the training originally, but we are only interested in males and females, so we reduced that to 2 categories. And in using this, we kept the weights, so we froze all the convolutional layers, and we only retrained the final fully connected layers with the mouse data. And our output were 2 scores, males or females, so whichever was higher, it was assigned to that class. And this is just an overview of the data we had. So we trained it on wild type mice, about 3,000, about 8,500 male, 1,500 female. We divided our data into training images, validation images, and test images. Validation images were used to prevent overfitting. And this is an example of the images that we used. And for our results, when we applied the model to the test data, unseen test data of the wild type mice, we had 98% accuracy. And when we applied it to unseen data from the knockouts, that's basically our entire knockout set, we had a similar level of accuracy 97.5%. And in particular, we are going to be looking for it later on in the number of false negatives. So we've restricted this presentation to just the males. So the number of males that were predicted as females is going to be important later on. In trying to determine what the model was using to make its predictions, we used 2 methods of visualizing the inner workings of the CNN. So the first one was occlusion sensitivity, where you take an image that the model works on properly, so it classifies it properly. You occlude a part of the image and pass it through the model and see if the model gets the classification right or wrong. And by moving this occlusion over the whole image, you basically can get a map of where the model loses, what parts of the image are important for the model to get the classification correctly. So we applied this, we moved it all over. So this is a 30 by 30 pixel block, and we moved it all over the image, and we did that for 100 images, and we built this map which showed that the chest area and the pelvic area were the areas that were most important. And we performed this again using another method, because occlusion sensitivity means that it's quite intensive, because you have to raster it over an image and then you have to repeat that for a number of images. Whereas there are other methods. In this case, it's an adaptation of a method by a group led by Zhu et al., where they use class activation maps. So they take the output of the image and they work backwards, and they use it to weight the final convolutional layer, and then they highlight the parts of the convolutional layer that are important in getting the result that you've got. And so in our case, we've done it, and we have this map, this class activation map here, and we superimpose that on an actual image that was used on the input, and we get something like this, which is the map over the image. And so this is applying that to our data set. So we've just put 3 random females and 3 random males, and you can see that for classifying as females, the pelvic region is important, and for classifying as males, the chest region is important. So if you combine these 2 together, you can see that the results of occlusion sensitivity and classification maps are consistent. So we looked at the 110 images that had false negatives, and it turns out that they were only from 87 lines, and we ordered them by the number of misclassified per line, and these are the ones that had more than 1 per line. And you can see that in the case of DUX02 TME, these 4 have a higher than expected number, which does not seem to be due to chance. So we looked at these 4 in more detail, and we also looked at some where there were no misclassifications. And you can see, so the green are the female scores and the blue are the male scores. But given that all of these are males, you'll expect the male scores to be higher, whereas you can see in the cases of the ones that were of interest, you have an inversion here, and then you have a great degree of overlap. And these are the class activation maps of the DUX02, and you can see that they're all similar to females, except this one, which is similar to a male. We took this, and these are the just normal x-rays, and you can see this is a male, a wild-type male, random wild-type female, and this is the DUX02, and it's more similar to the female. And then we took this to the human experts, and then they saw that there was at least 1 or both testicles missing in the x-ray images, or if present, they were small for the ones that we identified. And the conclusion was that this method is useful in pre-screening, and we have to do some more work on on finding out about why the chest area is important. And these are our acknowledgements. Thank you very much.